Hello and welcome to a presentation on the genetic disorder and disease cystic fibrosis. So before we get started here, I just wanted to mention that the reason why I chose cystic fibrosis is because it does run in my family. No one in my family has been identified as a carrier or as affected by the disease before my uncle did about 12 years ago. He actually did not exhibit some of the major symptoms that are commonly associated with cystic fibrosis. He had certain rare cases that didn't show up until certain events and later in life. I'll go ahead and explain those in further detail as we move through the slides and get a better idea for the disease. So I'd like to start first by giving an overview of what cystic fibrosis is and how it got its name. Cystic fibrosis was first discovered in the 1930s, although it likely had existed through human history. The term cystic refers to the development of pancreatic cysts and cysts in other areas where ciliated epithelial cells form mucous membranes. The term fibrosis then refers to the scarring and thickening of these mucous membrane tissues, often causing irritation and further complications. And then just for a little bit of background here, this disease is relatively rare, though for perspective, the CDC estimates that there are around 35,000 affected individuals living in the United States today. So here are some of the most common signs and symptoms associated with individuals who are greatly affected by cystic fibrosis. They generally include blockage of the airways due to a thickened mucus buildup, as well as inflammation of the lining itself, causing the difficulty to breathe. Also common to these patients, that same mucus blocks the passageways that pancreatic enzymes are excreted. Therefore, individuals are commonly malnourished and can't digest food. At various places in the body where the mucous membranes exist, infection is also common. So it's important to remember that individuals affected with cystic fibrosis are infected in areas where mucous membranes are present, and this includes a number of organs, more than just the lungs and the pancreas. So here we can see the inclusion of the stomach and the esophagus, the spleen, the liver, and other areas as well. And I'd like to point out that infertility is actually the way my uncle figured out that he had cystic fibrosis. They were trying to conceive my cousin about 12 years ago, and he was unable to do so because his vas deferens, which is where the sperm leaves the body, had actually become clogged and inflamed over time, and he was not able to have children at that point. So here we see the two individuals that were able to determine the gene location on chromosome 7. Francis Collins at the time was the director of the National Human Genome Research Institute, and Lapchi Sui, I hope I'm saying his name right, was a Chinese-born Canadian geneticist, and it was at his lab in Canada that they were able to find the gene location. They did it by observing linked markers inherited in families carrying cystic fibrosis, and they observed the recombination frequency to develop a measure of map units and estimate where the gene was. They then used DNA cloning and bacterial transformation to confirm the location of the gene and observe the correctly produced protein they were looking for. The first identified gene was a mutated gene, obviously, and it was delta F508, which is now called F508del. So in this slide, we'll take a look at the protein function. And this protein, the cystic fibrosis transmembrane conductance regulator, is the functional protein that's produced by the gene they identified. If we look at the image down here, we see that in normal airways, these epithelial cells that are ciliated have the attached protein to the plasma membrane, and it serves to pump chloride ions across the membrane. It's not actually the chloride itself that causes the thinning of the mucus. It's the fact that when chloride is moved to the extracellular space, it causes a hypertonic solution where water from the inside of cells will move to equalize the gradient by osmosis. So we see here the cilia is floating in water and the mucus is hydrated enough to be thin. Here it's compacted, the cilia that is, and the mucus is thickened. We also see in the image that the CFTR protein is not binding to the cell membrane. This isn't in all mutations, this isn't the case. And as we move to the next slide here, we see various mutations. So here in the image, we see this is the class of the particular mutation. And in some, it's the case where the protein's not created at all. In others, the protein isn't folded correctly and gets destroyed. Others have the creation of a protein that has a smaller half-life, so it's not as stable. But as you can see, here's our delta F508 in the image, and this is the most common form of cystic fibrosis, individuals that have 2 copies of that mutation. There are also 2,000 different mutations that have been identified that obviously are not included in this list, and each will have various detrimental effects to the affected individual. My uncle is actually known to have 1 copy of the delta F508 and another copy of one that's not on this list and one that he wasn't able to provide to me. But it was the combination of the two that didn't give him severe effects, but did cause the closing of the vas deferens throughout his lifetime. In this slide, we see the inheritance pattern, and conveniently enough, for cystic fibrosis, it is a pattern of simple recessive Mendelian inheritance. So here we see our 2 heterozygotes, the mother and father, producing a 1 to 2 to 1 distribution in the offspring. You do have to have 2 copies of cystic fibrosis in order to develop the effects. So here we see the cases for genetic testing, and to date, all 50 states require cystic fibrosis to be tested for in a newborn screening. This is done by a blood sample in infants, but for individuals that are looking to have a carrier test as well, the sample of DNA is taken and it's analyzed in the same way. In standard testing procedures, such as with the newborns, the gene is analyzed for a set of 23 common mutations set by the ACOG, which is American College of Obstetricians and Gynecologists, and the ACMG, which is the American College of Medical Genetics. There are over 2,000 various mutations that can be identified for the gene, although this is very rare because it's generally expensive. And this is actually what my aunt opted for. So since they were going to have to go through in vitro fertilization for my cousin, they decided that since my uncle was a confirmed case with 2 alleles, that my aunt should be tested, and they ended up spending a considerable amount of money to survey her gene for these mutations, and it turns out that she did not have a mutation for cystic fibrosis. So although genetic testings are pretty accurate, they can be inconclusive as far as determining whether the individual will develop cystic fibrosis in their lifetime. So for this reason, they use a sweat test, which is considered the most accurate test for cystic fibrosis. And in this test, they use a chemical pilocarpine and electrical stimulation to induce sweating of the skin. And in this image, we can see that happening in the top image. And then they go back and collect the sweat and test it to see the presence of chloride ions. A measurement of 60 millimole per liter of chloride is considered a positive test for cystic fibrosis. So for individuals that are affected by cystic fibrosis, the general prognosis includes a average life expectancy of 37 years, according to the University of San Francisco. This is a relatively significant advancement because in history, individuals normally didn't live to adulthood with cystic fibrosis. So with the new testing methods we have, we can implement therapies early on and allow individuals to live longer into lifetime as well. It is also speculated that with the new drugs that we've been researching, that individuals that are born today with cystic fibrosis may live into their late 40s and even longer. So when reviewing therapy methods, I did get the chance to speak to an occupational therapist. She's a friend of the family, and She worked in the city of Chicago at a pediatrics clinic and is now working in an adult facility in Marin, California. So, for physical therapy methods, it's important to have lung strengthening and esophageal strengthening for breathing and swallowing, basically eating. It's really important that individuals don't have food go down into their lungs because infection is incredibly dangerous for those that are affected with cystic fibrosis. Other therapy methods include the implementing of the high frequency chest wall oscillation vests, and you can see the individual on the right. He's living into his late 40s here, but is providing percussive motion to break up mucus in his chest, and it does so on the chest and on the back, which you can't see as well. I also thought it was interesting that in every article that I read, they adamantly refer to it as the vest, probably indicating that they have some love-hate relationship with the device. Other treatments that are very common in individuals with cystic fibrosis include the need for antibiotics. So a lot of times these antibiotics will be administered intravenously or otherwise through medications. Another common way to treat cystic fibrosis is like what you see in this young man here. He's using a nebulizer machine of hypertonic saline solution. So when the saline gets into his lungs, it creates that condition of hydrating the mucus as well as the saline causing the cells themselves to move water across the cell membrane. They also use common bronchodilators, similar to asthma patients. They also use albuterol. And then it's also important that individuals take pancreatic enzymes because they won't be excreted by the pancreas, and supplements of vitamins and nutritional eating as well. So the last set of treatments we're going to look at are drugs that have been developed, and these are based on what is termed SMIs, or small molecular intervention drugs. And the most commonly prescribed drug in America today is this drug called Trikafta. The tri refers to it is a cocktail of 3 different drugs that modulate the protein. So this drug is commonly administered for our most common mutation, the F508del mutation that we talked about in previous slides. And it actually serves to stabilize the protein after it's created. So in the F508del protein, there's actually a 3 base pair deletion that causes an improper folding of the protein and causes it to denature and be destroyed by the cell before it can function. These 3 protein modulators serve to fix that process and allow the protein to be attached to the cell membrane and function properly. In clinical trials with this drug, the 3 modulators showed an increase in CFTR function, that's our protein, decrease in measurable chloride in sweat, which is our sweat test, and increases in body mass index, showing overall health of individuals. The problem with these drugs is that, according to an article, Trikafta treatments annually cost an estimated $311,000 a year. So for most individuals, that's inaccessible and just not realistic for insurance companies either, unfortunately. So in the future, there have been proposed methods to better understand these protein modulators and develop new drugs, as well as develop SMIs, or these small molecular interventions, for other mutations as well, because not all mutations behave in the same way. So hopefully in the future we'll continue to progress and develop new treatments for cystic fibrosis. I'd like to thank you all for listening, and here are my works cited.